Hello, and welcome to our presentation on methods of cell lysis. Our group, Lauren, Kenzie, Max, and Benji, will guide you through 4 distinct techniques used to disrupt cell membranes. You will notice that for each method we have included a visual demonstration that we recreated using gel beads and popping boba to simulate cells. We will now explore the mechanisms, procedures, and specific trade-offs of each method. First is the freeze-thaw lysis method. In this method, cells are placed in a lysis buffer and submerged in an ethanol or dry ice bath. As they freeze, ice crystals physically puncture the cell membrane. The sample is thawed to let materials leak out, but a single cycle is insufficient. The process is repeated 3 to 6 times to fully rupture the cells, before centrifugation separates the debris. This method is inexpensive, simple, and gentle enough to preserve biomolecules. However, it is time-consuming due to the multiple cycles, less effective for tough bacterial or plant cells, and risks damaging temperature-sensitive molecules during the thawing phase. Next is the mortar and pestle method, the industry standard for processing hard tissues and plant cells. These samples have tough cellulose walls that chemical methods often cannot penetrate. The tissues are snap frozen in liquid nitrogen, which renders them brittle for easier crushing and keeps them cold to prevent heat damage to proteins. They are then ground into fine powder to physically shear the membranes, then reconstituted in a lysis buffer. This method is highly effective for hard tissues and offers flexibility, allowing you to use any specific buffer of your choosing. However, it is labor-intensive because it is done by hand. It can also lead to reproducibility issues between samples, and there remains a risk of degradation if the sample is not kept sufficiently cold during the grinding process. Third is the French press method, a method where a cell suspension is pressurized to thousands of psi and forced through a narrow orifice. The sudden pressure drop upon exit creates an extreme shear stress and microcavitation in the cell. This creates a highly turbulent system quantified by high Reynolds number, which effectively rips the cell open. The French press is effective for bacteria, scalable for large volumes, and requires no detergents. The main limitations are that the equipment is heavy and expensive. The process also generates a significant amount of heat that can harm delicate cells, and it may struggle with extremely tough fungi cells unless very high pressures are used. Finally, we have the bead beading method. This involves mixing cells with small ceramic and glass beads in a sealed tube. The tube is placed in a high-speed shaker where the beads collide violently with the cells. This mechanical impact is strong enough to rupture even tough membranes, like yeast and fungi. The bead beading method is fast, cost-effective, and chemical-free. However, the heat generated can damage sensitive molecules, so samples are usually kept on ice. Additionally, if the shaking speed is too high, there is a risk of shearing the DNA, and the process also produces fine cellular debris that can complicate downstream processing. To wrap up, we will have a short quiz covering the information we just went over and the concepts that were in our slides.
Stop running for me!
Thank you for your time, and here is our reference list.